talk of this session and the last talk of this year's Brain and Mind Symposium. I am very happy to introduce Dr. Benedikt Berninger. Dr. Berninger studied biology at the University of Munich. After a PhD on activity-dependent neurotrophin regulation in the laboratory of Hans Steinen, he joined Mu-Ming Pu's lab at the University of California, San Diego to investigate rapid action of neurotrophins at synapses and growth cones. Following his return to Munich, he became fascinated by adult neural stem cells and the neurogenic fate potential of glia. Working with Magdalena Goetz at the Stem Cell Institute, Helmholtz-Zentrum München, and the University of Munich, he discovered the proneural factors NeuroG2 and ASCL1 as key reprogramming factors for glia to neuron conversion. In 2012, he was appointed professor of physiological chemistry at the University Medical Center Mainz, and in 2018, he became professor for developmental neurobiology at King's College London. Since 2021, he has also led a satellite group at the Francis Crick Institute. His research covers a broad range of topics, from molecular mechanisms of neuronal reprogramming to circuit remodeling. His talk today is titled, Engineering New Interneurons in the Cerebral Cortex in Vivo. Dr. Berninger, welcome. Thank you for joining us today, and you can begin whenever you're ready.
So yeah, thank you very much for the introduction, and thank you very much for having me in this really interesting symposium. And I share my slides. I hope this works now. I hope you see my slides.
Yes, it's
yes.
we can see your notes.
We see your notes at the moment.
Okay, then this is the wrong side. How do I do that? It's not the notes, it's the next slide, I think.
Yeah, that should be it. No.
No. It's strange. You see what
Display settings. It's on the display settings on top.
Upper left.
What did you say?
On the upper left on the display settings.
Right here. You should be able to click on display settings.
Okay. Swap. Okay, brilliant.
Yes, there it is.
Okay, there were some slight challenges towards the weekend, so, but thank you very much for helping me overcoming that. So yes, let's get started with the talk. So the topics that I want to cover in this presentation is what is actually the rationale for trying to or aiming at converting glia into neurons, and what are the key challenges to actually achieve this in vivo context. And a big question that actually is becoming ever more important is how good is actually our evidence that at least some of this reprogramming of glial cells into neurons is actually possible in vivo and it's really authentic. And ultimately, or one question that I want to address towards the end of the talk is, what are our perspectives of generating subtype specific neurons, such as interneurons, through reprogramming strategies? So the problem is depicted here. Essentially, if there's any insult to the brain, injury or a disease that results in death of neurons, we know that unfortunately this process is not happening. So there's no spontaneous regeneration occurring. And this has been already been known since a long time. For example, Santiago Ramón y Cajal made this very important or interesting statement that he said everything may die, nothing may regenerate, but it is to the future of science to overcome this harsh decree. Now, we are trying to find strategies that would eventually enable the regeneration of neurons that are degenerated, or maybe on a more subtler way to think about strategies to adding new neurons to diseased brain circuits. And the idea is based on a dream, which I call our working dream, namely the possibility of converting local endogenous glial cells, such as astrocytes, but also other type of glial cells, into neurons. And this slide just depicts actually the daunting task if you think that you would really want to remodel or regenerate circuits, which are of course composed of a plethora of different type of neurons. So at this point, I think we are far away from really having this goal very concretely in front of our eyes. And so what we really have to do at this point is to have a proof of principle studies where we would examine whether it's possible actually to convert glial cells into somewhat specific type of neurons as a proof of principle. So we have started this more than a decade now ago when I was in Magdalena Götz's lab. And at that point, we really focused on in vitro systems, culturing astrocytes from the postnatal cerebral cortex and challenging these astrocytes with a panel of transcription factors, of which we knew that they play key roles in steering cell fate in the developing cerebral cortex, such as neurogenin 2 or MESH1, which we now refer to as ACIL1, but also DLX2. And indeed, in vitro systems, we could see that when we introduce these transcription factors, cells eventually turn from their glial identity into a neuron. You see this here in a time-lapse study over the course of several days. A cultured astrocyte eventually turns on the expression of this expression vector, which encodes a reporter, but also neurogenin 2. And over the course of a couple of days, the cell turns into a neuron. So demonstrating that at least cultured astrocytes could be converted into neurons. Now, just like a brief excursion, but I think with important implications, I want to discuss the question of how this is actually achieved. Now, there's two possibilities, in fact. So we refer to this kind of conversion of cell fate as direct reprogramming. And the term direct, I think, is a little bit confusing and maybe sometimes even misleading. So in most of the cases, direct reprogramming means that, as you have seen in this experiment, that a cell identity a switch in cell identity is occurring in the absence of cell division. This doesn't mean that cell division cannot occur, and I show you an example just in a moment where cell division occurs, but it is not a prerequisite to happen. So, but direct on a first glance actually seems to suggest that there is a cell of a given identity that then loses its original properties and acquires the properties of a new identity. For example, you start with an astrocyte and that cell turns into a neuron. That would mean that you lose the expression of astrocyte-specific genes and then gain at the same time the expression of neuron-specific genes. This has been discussed whether this happens in this way or in another way has been very nicely discussed by Samantha Morris, who actually proposes that in many cases the reprogramming works actually more according to this model, which is referred to the pioneer factor mediate conversion compared to the terminal selector mediate conversion. And in this case, what really happens is that the cell is not just directly converted into another cell, but is first acquiring a progenitor-like state, and from there then starts to differentiate into its new identity. And this video here actually shows something very interesting that may be suggestive of that indeed there are at least intermediate states in this reprogramming process. So it's not simply just losing its original identity and acquiring a new one, but that this goes through different steps. And in this case, you actually see that a cell is first dividing in this process, and then at some point, while one cell dies, the other cell starts to exhibit actually a morphology that is kind of reminiscent of multipolar progenitor cells, before the cell then starts to send like a process that could be an axon-like process and turns into a neuron. What I think is very remarkable and unique in this video, because we don't see very often cell division when we see this conversion process. But what is interesting is that clearly this process is a challenging one. So one of these two cells dies, and it happens just at the transition between the more flat morphology and the moment where the cells are turning into this multipolar progenitor-like state. So you see this maybe now when we run this movie again. So at this point, when the other cell died, this cell is turning into this multipolar progenitor state, which suggests to me that perhaps there is a key hurdle to be taken in order to manage this conversion process. We have been trying to analyze this in a slightly different system in molecular terms, and this was work that was actually spearheaded by Marisa Caro, who recently started her own lab at the University of Erlangen, and she was actually working with cells in culture that have an expression profile very similar to pericytes. So we think they are brain pericytes following dissociation of the brain tissue, and these cells were derived from human patients. So this connects somehow my talk here now to David's, even though we do something really awful here, namely convert these really important cells into neurons. But that's not, in this case, the ultimate objective, of course. So what we wanted to understand here was, so what Marisa found was that when she overexpressed ACIL1 or MESH1 together with SOX2, she very efficiently got conversion of these cells into neuron-like cells. And to understand what actually happens during this process, we teamed up with Barbara Treudlein and Greg Camp, really brilliant scientists who were very early participating in the search of single-cell transcriptomics of neuronal cells. And this is, of course, very early single-cell data. So nowadays you would expect tens of thousands of cells, but this was actually a really revealing experiment, even though we had only a couple hundreds of cells in this experiment. So what we found was that when we overexpress ACIL1 and SOX2 in pericytes and follow then the change in the transcriptome by arranging these transcriptomes in pseudotime, we see that, in fact, pericyte genes are downregulated while eventually neuronal genes are upregulated. But there's actually a broad range of genes, and there are some examples here, which exhibit a dynamic regulation in the sense that you switch these genes on and then downregulate them later as the cells mature into neurons. And these genes are, it's interesting, I cannot go into detail here for time, but what we saw that the induction of these genes seems to be really critical for successful reprogramming to occur. So cells that fail to induce this set of genes wouldn't reprogram. And when we look where are these genes normally expressed, we find that they are highly enriched in radioglial cells, so in neural stem cells. So what this suggests to us is that as these cells progress from a pericyte into a neuronal identity, these cells actually run through a stage where they resemble neural stem cells before they then differentiate into neurons. So this so-called direct reprogramming nonetheless involves intermediate states that are sort of determining the final outcome. And just to highlight one particular aspect of this, that this might be biologically important, is we found that at this state where the cells are in this more neural stem cell-like phase or stage, we see that many components of the notch signaling pathway are highly expressed. We see that the ligands are expressed, we see that the receptors, notch2 is expressed, and we see some downstream target of the notch signaling pathway expressed. And we found that when, so Marisa, when she did the experiment, when she blocked notch signaling in these cells, she got actually more cells differentiating into neurons compared to control cultures. And this actually suggests that indeed, during this transition phase, cells behave almost like endogenous neural stem cells in the sense that they receive lateral inhibition. So as cells differentiate, they will present the ligand of the notch signaling pathway, telling to the other cells not to go ahead with differentiation. And we see this behavior also in this in vitro system, of reprogramming pericytes into neurons. So this indicates that indeed understanding the intermediate states in this process is really critical, important for understanding the biology of these identity switches. Now, for the rest of the talk, I want to go to actually the situation in vivo and to the main goal in the lab to see whether we can actually convert endogenous glial cells into neurons, such as astrocytes, but we are also interested in the potential of oligodendrocyte progenitor cells to convert into neurons. And you may have seen many studies in the recent past, which actually came with really amazing claims about what is possible. And I just want to highlight that one has to be really careful here in getting the methods right to study this process. So what has been found is that many studies have been using adeno-associated viruses because they turn out to be very powerful in infecting really broad range of brain areas if you inject them into the brain. And they have been used by several groups to convert astrocytes into neurons. And the amazing thing was that these neurons really resembled the neurons that you find endogenously in the brain. And this was in cortex and also substantia nigra and the retina. However, it turns out that there's a technical flaw in the system, which actually is not fully understood. So AAVs somehow, even though they can be tailored to be expressed in glial cells initially, depending on exactly what you put into these AAVs, seem to be eventually become active also in endogenous neurons. And then you may believe or may think that these were actually induced neurons, but in fact these are endogenous neurons. And if you're interested in this problem here, I want to highlight this recent publication from our colleague Chun-Li Zheng from the University of Texas Southwestern. And I think this is a very important study because I think it sort of resets us a little bit back to the stage where we understand that cell identity is not that easily dealt with. No? I want this just give you as a background to our own attempts of reprogramming glial cells into neurons in vivo. And so the model that we use is maybe more humble than the adult cerebral cortex, is the P5 cerebral cortex, because at this stage, glial cells such as astrocytes and oligodendrocyte progenitor cells are still highly proliferative, and you can target them by retroviruses like the mouse Maloney leukemia virus, which only successfully transduce cells that are in proliferation. And I think this is a very important constraint that helps us to secure the glial origin of the cells that we target in this process. So the idea is to inject directly retroviruses encoding these reprogramming factors and to see whether we can convert them into neurons. And this is the work of a team in the laboratory, spearheaded particularly by two really talented postdocs, Sophie Perron and Nicolas Marichal, and with contributions, very important contributions from different PhD students. So Chiara Galante did some part of the work here. She already finished her PhD, and there are two additional PhD students working on this topic, Ana Beltran and Laia Torres, who work on the question, can we convert in vivo glial cells into neurons? Now, the first thing that we tested was, do we indeed transduce glial cells when we inject these retroviruses into the brain? And the first analysis that we did was to see whether, what glial cells, cell populations are targeted, and the vast majority, at least when you inject a control virus, is cells that express either GFAP or SOX10. So suggesting that more than 90% of the cells are either belonging to the astrocytic or oligodendroglial lineage. There are very few cells that will be expressing macroglial markers, suggesting that it is not a situation, at least when you have an acute injection of the virus, that you have massive macroglial division, and that would eventually lead to subsequent transduction of these cells. There are 0 actually double cortin positive cells, suggesting that at this point there are no more progenitors that give rise to double cortin positive cells later on, and that I think is very important. Now, when we apply ACL1, which is one of the preneural factors that is a very potent reprogramming factor in vitro, in the in vivo context, actually the outcome first is quite remarkably disappointing. Namely, we see very little, if any, change compared to a control virus. So if we inject these kind of retroviruses that I mentioned that require proliferative cells, we see that we get quite nice transduction, but we do not see massive changes in cell identity. So there's hardly any cells expressing double cortin. Now, we knew from work from Carol Schurman's, but also Anna Philpott so Carol Schurman's mostly on ACL1, but also Anna Philpott on neurogenin 2, that these preneural genes are regulated by phosphorylation. And phosphorylation actually promotes a glial program, while a less phosphorylated state is leading to neurogenesis. So ACL1 can be phosphorylated on 6 serine residues, and the idea is that perhaps perhaps the lack of conversion was really due to simply to the fact that these serines would be phosphorylated, and therefore we don't get neurons but glial cells. So what we did was we got from Carol Schurman's a mutant in which these 6 serine residues were mutated such that they are now alanines, and we refer to this as ACL1 SA6, and we place these variant into our retroviruses and then assess whether this would have a change on the outcome. But the result was still extremely disappointing. So when we expressed wild type or this mutant, we hardly found any change in the levels of double cortin positive cells. So this suggests that, in fact, ACL1, at least alone, is not capable, even in its more neurogenic variant, to convert the glial cells into neurons. And I think this is already a very important point, because it indicates that it is not just that these cells just need a kind of kick and they would just go down the neuronal trajectory. They actually want to stay glial cells, and you don't overcome this hurdle just by one transcription factor. Now, we could build, however, on a very interesting observation made by Sergio Gascon while he was a postdoc in Magdalena Götz's lab. And what he found was that when he overexpressed BCL2, this would promote, in fact, the conversion in vitro and also in vivo into neurons. And the mechanism that he proposed is that BCL2 was, in fact, acting as an anti-ferroptotic molecule. So ferroptosis is a form of cell death that is induced by reactive oxygen species. And the mechanism is not exactly the same as apoptosis. So this is not just blocking apoptosis, but more specifically this process. But I have to say, I believe that probably the role of BCL2 in this process may be more complex. Whatever the mechanism, what is really clear is that when we overexpress now these factors, ACL1 or the mutant one, ACL1 SA6, together with BCL2, we do see clearly the induction of double cortin positive cells in the cerebral cortex. And this is 12 days after the injection into a P5 mouse brain. So this is indicated. So at this stage, there's no double cortin positive cells, at least cells that express double cortin at this significant level in the cerebral cortex, and you also see that these cells here are actually clearly looking neuronal. What became also very clear from these inspections is that the wild type is indeed much less potent than the mutant ACL1. And so this is another example where we actually combined ACL1 and BCL2 in a single construct. And here you see that most of the cells at a later time point stay, many cells stay in fact in their glial lineage when you overexpress the wild type ACL1, while if you use the mutant version, the majority turns into neuronal cells, and this is quantified here. So there's still quite a similar range of cells expressing double cortin between the mutant and the wild type and the mutant, but there's significantly more cells inducing the more mature neuronal marker NeuN when you overexpress the mutant. So the mutant clearly is improving the outcome of reprogramming, but you see that you need clearly here a cofactor, as these factors alone are in fact very little have very little potency to induce conversion. Something which we are, this is just like work in progress, what we are currently trying to do, because I think this is very important to understand how expression levels correlate with reprogramming efficiency. And we are haunted by the fact that many of these proteins that we use, like ACL1 or neurogenin 2, they are not very good antibodies against these, but the RNA-scope technology allows us now to address this in a more quantitative manner, and we are just addressing actually whether our cells indeed express, at least on a messenger RNA level, the factor that we overexpress. And I think this is very nice to see that indeed this is the case, and we would like now to see, can we actually relate expression levels to reprogramming efficiency. I told you that the viruses that we use indeed target proliferative cells, in the sense that stable transduction requires, in fact, the disassembly of the nuclear envelope in order to get the retrotranscribed viral genome integrated into the host genome. And so if this is true, what you would expect is that you would actually have to be able to see that the cells that are virally transduced also incorporate, for example, a thymidine analog like BRDU. And so what Sophie Perron did to test whether this is actually true, she administered BRDU before she did, or at the time, around the time when she induced or injected the retrovirus. And what she sees is that indeed, both for the wild type and the mutant, we see that the vast majority of cells that we obtain that turn into neurons indeed also incorporate BRDU, indicating that indeed these cells that we label here, they are not endogenous neurons, because if you look at the endogenous neurons, these would actually no longer incorporate BRDU. So it's the BRDU incorporating cells are indeed the retrovirus transduced cells are indeed those cells that are incorporating BRDU, and the endogenous neurons at this stage no longer are of any proliferative nature, and therefore fail also to be transduced with the retrovirus. Maybe side conclusion that I want just to make is that there are cells that reprogram and there are cells that fail to reprogram. And one thing that is interesting, if you quantify the level of BRDU labeling in cells that successfully reprogram versus those cells that fail to reprogram, suggests that indeed there's higher BRDU levels accumulating in the converted cells, and that is consistent with the idea that while you convert, actually cells are exiting from cell cycle or kicked out of cell cycle, while the cells that fail to reprogram continue at this stage still to undergo some rounds of cell division, and this results in a reduced or in a dilution of BRDU, as cells incorporated the BRDU but then lose this with several rounds of cell division. And this is suggesting that indeed the reprogramming might in many cases lead to direct exit from cell cycle and therefore also be a direct reprogramming in the sense of not involving proliferative steps after the process has been started. Very important, because this was one of the main problems found with many of the AV studies, is that when Tun Lee and his lab assessed genetic fate mapping to see whether indeed cells that were genetically fate mapped as glial cells and then were transduced with AVs, that these cells wouldn't actually correspond. So you got the glial cells labeled, but the neurons were actually not exhibiting the glial fate map. So we did this experiment to see whether indeed when we did genetic fate mapping and then transduce cells with retroviruses encoding the reported gene, whether these would actually, whether we would see cells that can be fate mapped. And here you see a clear example of a cell that appears to be of an astrocytic origin. So in this case, we use a mouse line that expresses chief recombinase under control of a astrocytic promoter or a regulatory element of the mouse GFP locus, and combined with a mouse line that has a GFP following recombination. And what you see is that here lots of glial cells are recombined, but we have a glial cell that was transduced with, in this case, ACL1, SA6 together and with BCL2. And then we see here that this cell indeed expresses double cortin and shows the fate mapping, so indicating that glial cells such as astrocytes indeed can be reprogrammed into neurons. When you quantify this, we see that about a quarter of the cells belong to this category. We see another quarter or maybe a fifth of cells that do not express double cortin in these experiments, but we need to quantify that a substantial proportion of these cells expresses NeuN and no longer the immature marker. So that maybe there's a substantial amount of cells here that are reprogrammed, but already a step further compared to this stage here. Now there's also about a quarter of cells that are targeted by the virus and were not fate mapped as astrocytes and may have or at least didn't induce double cortin. And there's a quarter of cells that is clearly not fate mapped and yet start to express double cortin. And this is something which we have to confirm, but we believe that this is most likely cells of the oligodendroglial lineage. So we are currently conducting experiments where we do the analog with with regulatory elements active in oligodendroglial progenitor cells to see whether we indeed can confirm that this quarter here can be attributed to conversion of oligodendroglial cells in this model. And just as other important point here, that at least some of these cells seem to make it for a while. So this is an example of a converted cell that could be found 3 months after induction. And yet I want to highlight, despite the fact that this cell actually looks pretty nice, if you would compare this to an endogenous neuron, these cells are clearly not of the same degree of complexity and often not even having cell somata of the same size as endogenous neurons, which indicates that indeed the cells that we obtain in this model are not yet perfect neurons, which also indicates or argues along the line that this is really authentic cell fate conversion process, which however may be imperfect. So this is essentially the conclusions from this first part. So we can actually induce neurons from proliferative glia. At least some of these are of an astrocytic origin. These cells pass through an immature neuronal state that is characterized by double cortin expression. And very importantly, we find that an ACL1 variant that is phosphoserine deficient is more powerful than the wild type ACL1. Now, can we reprogram in the adult brain? And here we did the following experiment because we really wanted to be on the safer side using our retroviral expression system. What we performed was a stab wound injury, which induces proliferation in these in the region of the lesion, and then transduced using the same retroviral set proliferative glia 3 days after induction of this injury, and then analyzed at subsequent stages how these cells behaved. And when we inject the control virus, we see that there are cells that can be transduced indeed, and many of these cells, or the majority of these cells, clearly exhibit endogenous glial morphologies and do not express any NeuN, which would be indicative of a neuronal identity. Now, here the difference between the mutant and the wild type and the mutant ACIL1 is even more clear. So here you see what happens when we overexpress a wild type ACIL1 together with BCL2, and here you see what happens when we overexpress the mutant ACIL1. So the wild type doesn't seem to result in much of a conversion, and it looks like that cells actually continue for a while to proliferate, while here the mutant ACIL1 triggers neuronal differentiation together with cell site exit. And indeed, we can see that many of these cells here, they start eventually to express the more mature neuronal marker NeuN, and you see that this is actually the difference between wild type and mutant is very significant. So that there's a dramatic difference, but there's of course also a difference in cell numbers, as you could appreciate from this. So we get many few cells, but and we need to find out whether this is indeed due to the fact that this is due to cell cycle exit, or whether there's actually some loss of neurons as they differentiate in this adult brain. So this would be just like a minor conclusion here along the path, again highlighting the fact that the mutant ACIL1 is more powerful in inducing reprogramming, but very importantly also that reprogramming can also occur in the adult cerebral cortex. And we would like now, of course, to know what glial types were responsible for that, again, whether this is astrocytes or oligodendrocyte progenitor cells. Now I want to come to perhaps one of the most surprising findings that I want to discuss with you, namely, can we induce some kind of subtype specific properties? And here I show you a patch clamp recording from a brain slice obtained from a mouse that got injection of ACIL1 and BCL2, and Nicolas Marichal made this patch clamp recording in this slice, and he found that the cell indeed exhibited some electric excitability, but everyone would be very disappointed with such an outcome. And in many cases, and particularly also David would stress this, this is something that you could easily imagine to find in an oligodendro glial cell or a progenitor already. So this is not really a clear example of acquisition of a neuronal identity. Now, when we did the same experiment with the mutant variant, the outcome is quite different. And what we see here is that, in fact, these cells now can spike at much higher frequencies, but what was even more impressive, a quite significant amount of these cells can now spike at frequencies that correspond to classical fast spiking interneurons. That means above 100 hertz or more. And this is quite remarkable because this enrichment of cells that express this, exhibit these electrophysiological properties, you would not expect this just by a random sampling of interneurons or neurons in general. But this seems to suggest that expressing this particular variant of ACIL1 in the context of a BCL2 overexpression at least activates some expression modules that are required to induce an electrophysiological identity of a fast-sparking interneuron. Now, interesting, we find that some of these cells are also present in layer 1. And this is interesting because normally these neurons are not found in layer 1. So this indicates a very important point for us, that these reprogramming event is in fact not activating the same kind of cell identity programs that you find in the corresponding brain area anyway. No, so if you would expect that these cells behave in a way that you get exactly the type of neurons that you find there otherwise, I would be actually a little bit worried, and I have been always worried about this when this was reported for several of these AV studies, that rather to find that actually the transcription factor can induce identities that do not correspond to what you find locally there under normal condition, somehow adds to the evidence that this is indeed an authentic conversion of identity in this case. This is just highlighting some of the differences between the wild type and the mutant variant. So while the wild type really exhibits very unimpressive electrophysiological condition properties, the mutant is clearly exhibiting a marked difference, and many cells exhibiting a firing range of fast-sparking interneurons. And consistent with this, action potentials are of course very brief in order to allow for this high-frequency firing. And also we see that overall these induced neurons that express the mutant ACIL1, they also are better synaptic targets for endogenous or host neurons, so neurons that make synapses onto these cells. And we find that indeed they receive synapses, but these synapses are again found more often on neurons expressing the mutant ACIL1. And consistent with this fast-sparking interneuronal subtype, we do see also expression of parvalbumin in at least a substantial proportion of the cells, so not in 100% of these cells, but in roughly 20%. There are also cells that are actually negative for this, just to show you the example that indeed not every of these induced neurons express parvalbumin. But what is interesting, we see also differences to the endogenous neurons. So when we again performed RNA-scope analysis, we see that the wild type cells rarely express any significant level of RNA-scope signal for the parvalbumin messenger RNA compared to the mutant one. And here we see actually many more cells expressing parvalbumin on the messenger RNA level compared to the protein level, but the levels are dramatically lower compared to endogenous neurons. So here you see endogenous PV neurons that express really by order of magnitude higher levels of this messenger RNA. So this suggests that our cells do acquire some of the hallmarks and features of PV interneurons, but we shouldn't equate them, and we should really avoid the danger of pretending that we have here found a way that expresses results in a parvalbumin interneuron. But what is interesting is that the strategy that we use is capable of activating some of the modules that are required or probably participating in the endogenous generation of PV neurons as well. So this is just like the slide that shows you that there's no PV staining in layer 1, while PV neurons occur all throughout the cortex. And this is, there was a slight problem in the organization of my slides. So this is the slide which shows that we can actually induce a PV phenotype in layer 1, where you normally wouldn't find this. And we also see that in this layer we find fast-sparking interneurons that you normally wouldn't find in layer 1. just a very final data slide that I wanted to show you, which is very interesting and which is maybe a little bit changing the gears here, but which is very exciting. So this is work that we performed in collaboration with the laboratory of Christoph Heinrich. So actually he spearheaded this, and this is his PhD student, Celia Lentini, and the electrophysiology was done here by Nicolas Marichal. And the experiment was the following. So Christoph was interested to see whether inducing neurons, interneuron-like cells, in the context of an epileptic hippocampus could indeed bring about some functional amelioration. And so what Celia did was to inject first carnic acid to induce here a focal epilepsy, a model of epilepsy of mesotemporal lobe epilepsy. And then a couple of days later, a reprogramming factor cocktail was expressed. So here, DLX2 was replaced by, or replaced here BCL2. So this was kind of a different starting point that Christoph and Celia took compared to our studies that were running in parallel. And also this is wild-type ACL1. But remarkably, in the context of this epileptic brain, where you get reactive gliosis, some of these, or quite a few of these cells turned into neurons. And so what we then did was to, and this was really remarkable because this happened, we did these experiments during the pandemic and discovering the consequences of Brexit at the same time. So so these mice were produced in Lyon, and then they were sent over to London, and Nicolas then prepared brain slices and wanted to test whether actually by inducing these induced neurons to fire, whether he could see responses in granule cells that would be some of the remaining cells. So most of the cells actually die in the context of this epileptogenesis, and particularly also the interneurons. So most of the interneurons die in this following carnic acid injection, but the granule neurons in the dentate gyros are resistant, and they are the most significant substrate of the epileptogenesis process. Now, Christoph and Celia had found that indeed inducing interneurons using these two factors allows for a reduction in the amount of seizure activity in the animals when measured 3 or 4 months later. So what Nicolas then wanted to see is whether we indeed can see synaptic transmission between induced neurons and granule cells. And so what he did was to do patch clamp recordings combined with optogenetics. And this is essentially what he did was to shine laser light on cells that were surrounded by, or shining laser light on the surrounding of cells that were surrounded by fibers that were GFP positive, because the channelrhodopsin that was expressed at the same time along with the reprogramming factors was targeted to the induced neurons. And indeed what Nicolas could see is that when he shined light on these cells, he could see synaptic transmission that could be blocked by GABAergic, suggesting that indeed these induced neurons can form functional GABAergic synapses onto granule cells, and perhaps through this mechanism contribute to a reduction in seizure activity. Okay, so a final set of conclusions. Again, it's interesting to observe that varying the or using different mutants of ACL1 can have different outcomes on the electrophysiological properties. And what we found really remarkable is that when we use the serine deficient variant of ACL1, we obtain actually quite a few of cells that express electrophysiological features of fast sparking interneurons. We see this even happening in layer 1, so in ectopic locations where you wouldn't find these neurons normally. And that suggests that at least to some degree, transcription factor mechanisms can prevail over the regional identity of the cells. I make a small question mark here because we do not know what is the origin of the cells. with regard to the electrophysiology, so whether the fast-sparking interneurons are of astroglial origin or oligodendroglial origin, and that may have some consequences on the way these cells respond to these factors. And finally, at least in some particular cases, like if you study this in an epileptogenic environment, the induced neurons can form functional synapses onto target cells. So with this, I want to thank the people who have done the work. So I highlighted particularly the work of Sophie Perron and Nicolas Marichal, and the work of Anna and Laia, who contributed to this project. And I thank the funding agencies, and I thank you for paying attention. Thank you very much.
Thank you very much for your talk. We have some time for questions from the audience now. So the first one is, the data show that not all astrocytes are converted into neurons after the expression of ACL1 and BCL2. Are there subtypes of astrocytes which are more prone to be converted into neurons?
I think it's an interesting question, but we don't know this yet. No, so I think there's the very first question that we have is actually, so the way that we do it is by using the retroviruses that target proliferative cells. And you could, of course, ask the question, can we also reprogram fully mature non-proliferative astrocytes? And this is, for example, a question that is at this point for me is still open. So the earlier work with the adeno-associated viruses seemed to suggest that this would be possible, because you could inject the AVs and you got lots of neurons, and it appeared that many of these neurons would not be labeled with BRDU, so suggesting that these cells had not been generated from astrocytes that had proliferated. But since this evidence is now somehow no longer convincing at this point, I think it might be very well possible that this fake conversion potential can be really observed in, or might be still more plastic in proliferative cells. And so this is number one, which we need to learn. But then, of course, and maybe just a very brief comment to finish this, of course, then it will be interesting to see whether glial cells that are proliferative because of different conditions, normal development versus reactive gliosis, whether they have different competence to be reprogrammed. That's totally open, and we would very much like to learn this. Yes. Okay.
Thank you very much. We have another question. How important the proliferation is in the conversion? Have you ever tried to convert astrocyte into neurons using genetically modified mice, not by retroviral injection? Is the retrovirus injection induced reactive astrocyte proliferation important for the conversion?
So this is a bit what I think I already addressed. We haven't done this. Would be still a very valuable experiment to do this actually through transgenesis. So having a possibility of having a mouse line where you could induce the transcription factors with doxycycline and do this at different stages. We haven't done this yet. This is a really nice experiment. My suspicion is that it becomes more complex or more difficult to convert fully mature astrocytes compared to astrocytes that are dividing. But it is still an open question.
Okay. Could the conversion of glia into neurons cause any short-term disruptions or imbalances in local networks? And if so, what kind?
Yeah. So, I mean, we just heard before the talk by David, who clearly showed how important not only, so astrocytes, of course, and other cells in the brain are like pericytes, and we totally underestimate their role. And so what we do not, the claim is not that we want to convert now all glial cells into neurons, because obviously that wouldn't work, and you would probably have the same amount of detrimental effects on network function compared to the disease state itself. on the other side, of course, this is proof of principle. So what we hope to become able is to understand how a fate of a cell in the brain could be converted, such that if we then would like to use this at some point in a therapeutic way to steer this process in a much more subtle and in a quantitatively defined way. Of course, we would also somehow hope, and that hope might be disappointed by the hope for the possibility that to some degree glial cell population can replenish, so that if you would convert a glial cell, that this converted glial cell can then turn, this could be replaced by surrounding glial cells that then would continue proliferating and maybe replace this cell. So this is something where we think that we need to go stepwise. We cannot address all these important questions at this stage, where we even, to my understanding, have not yet reached the point where we can say really astrocyte-to-neuron or glial-to-neuron conversion really cures a neurological or neurodegenerative disease.
Right. Okay. Thank you.
Thank you. I guess we can take one more question from the audience.
I think we have one. We have time for one more. Yeah.
Yeah. Is it possible to induce principal neurons?
Yes, I believe so. So if we use we can also, of course, we can use neurogenin 2 instead of ACIL1, and it turns out that this indeed makes a difference. So we see neurons that have morphologies that are going more towards pyramidal neurons. We see also that these cells often exhibit spine-like structures. So that seems to be possible. But again, we don't know to which extent these principal neurons are really making long-range projections and so on. This is something which I think still needs to be revisited. So there was reports in the literature that seem to suggest that, but we need to look carefully at how solid all this is and keep working on it.
Okay. Thank you again for your time. I think that's all the time we have for questions at the moment. So thank you again for your talk.
Okay.
And for joining us.
And thank you for having me.